Today we will discuss about the centriole. What is centriole? In the last class we have discussed about the microtubule, and in this part we will discuss more things about this centriole. Actually this centriole is made up of what? The centriole that is made up of microtubule. Okay. And what is the definition? These are non-membranous hollow microcylinder. These are non-membranous hollow microcylinder made up of microtubules. Okay, and when we see its occurrence, this is only found in case of your animal cell. This is only found in animal cell, and this is absent in plant cell. Okay, and as these are present in animal cell, these are occurring in pair, and they arrange perpendicularly, Okay, when we see the centriole, there is presence of two number of centriole. This is one centriole. Suppose these are micro cylinder, these are just cylinder like. Okay, so these are non-membranous structure. There is absence of any membrane, so that this is non-membranous hollow microcylinder. As they are very small cylinder, that's why that is microcylinder, hollow microcylinder. These are microcylinder. Okay, and made up of the microtubules. These microcylinder, these are made up of what? These are made up of the microtubules, and as you know that the microtubules are just hollow cylindrical structure, and this centriole is also hollow cylindrical structure, and there is presence of two number of centriole, and these centriole that are arranged in a pair. This is suppose another centriole. Okay, and both these centriole, they are arranged perpendicularly. This is two centriole, they are arranged perpendicularly. And this pair of centriole, that is called as what? This pair of centriole is called as diplosome. Pair of centriole, that is called as a diplosome. Okay, and around this diplosome there is presence of some amount of cytoplasm. Around this diplosome there is presence of cytoplasm, and this is the cytoplasm or semi-fluid substance around the diplosome, and this substance which are present around the to centriole, they are making a new structure. So this is the cytoplasm. Let's write this. This is the cytoplasm around diplosome. Okay, this cytoplasm, diplosome with that cytoplasm, okay, or this cytoplasm around the diplosome is also called as kinoplasm. This is also called as kinoplasm. So diplosome and kinoplasm, they are making the structure that is called as centrosome. So what is centrosome? When we consider this whole structure, this whole structure is called as what? This is called as centrosome. Centrosome, that is made up of two number of your centriole. This is centriole 1, this is centriole 2, and around the centriole there is presence of what? Cytoplasm, or that cytoplasm around the two centriole is called as kinoplasm. And these diplosome and kinoplasm, they are collectively called as what? They are collectively called as centrosome. Okay, and when we see more details about this centriole, let's see one of the centriole. One of the centriole, that is a cylindrical structure. This is a cylindrical structure, and in that cylindrical structure there is presence of microtubules. Suppose these are the microtubules. These are the microtubules, and these microtubules that are present here. This is one microtubule. This is another microtubule. Okay, this is one centriole. This is one centriole, and another centriole that is present perpendicular to this. So these are the wall of the microtubule. Okay, so these are arranged perpendicularly. Two number of centriole, right? So when we see its transverse section, when we took its transverse section, suppose we have to just go for this transverse section or outer part. Let's see here from the front view. When we see its front view, then we will go for the transverse section. The transverse section of centriole, it gives a cartwheel structure. It gives a cartwheel structure, and that cartwheel structure having microtubules. Okay, so what is cartwheel structure? when we took the transverse section of this centriole, then there will be appearance of a cartwheel. So our choka will have a structure creation about that structure is known as what? That the cartwheel structure of the centriole. Okay, so let's see the cartwheel structure of the centriole. Before that, let me show you a diagram, or that is the image that I have downloaded from the Google. Let's see this diagram here. Okay, let's see. This is the cartwheel like structure. So this part, so this is one microtubule, this is another microtubule, this is another microtubules, these are the microtubules. So these are triplet microtubules. There is presence of triplet microtubules. So how many triplet microtubules are there? There is presence of 9 triplet microtubules. This is 1, this is 2, this is 3, this is 4, this is 5, 6, 7, 8, 9. There is presence of 9 microtubules. Okay, and these 9 microtubules, but this is not microtubule, this is triplet microtubule. 3 microtubules, they are joined here and they are making a triplet microtubules. Okay, so when we see this transverse section, means we have to just cut this part, cut this section, and we have to see this from the front. Then how it will look? The transverse section, that will gives cartwheel structure. Okay, so let's see the cartwheel structure of this centriole. Okay, cartwheel structure of centriole. Okay, so before that, there is 3 microtubules and there are 3 hollow structure, 3 hollow microtubule that are getting joined, and after they are joining, they are making triplet microtube, or we call this as triplet 5D. Okay, suppose this is one microtubule, this is another microtubule, this is another microtubule. These microtubules are getting joined with each other, but out of these three microtubule. This is one microtubule. This is another microtubule, and this is another microtubule. Okay, so these three microtubule when joined, this is suppose A, microtubule A, this is B, and this is C. Out of these three microtubule, this is your triplet fibril. The A microtubule, that is complete microtubule. This is complete microtubule. But these two that are incomplete, these are incomplete. Why that is called as incomplete microtubule? Because when we see the microtubule in the last class, I am forgetting to discuss that section actually. What is complete microtubule? Actually the wall, this is the wall of the microtubule, this is wall, this is the wall of the microtubule. In that wall there is also presence of our dimer, alpha beta dimers, alpha beta tubulin proteins are there in that wall also. The pair of alpha beta globulin, or sorry, that tubulin proteins are there. So this is this part, okay, this one section. If we took this part, this is one, this is another row, sorry, this is column, these are column, these are another column, this is another column, this is another column. So these columns are making what? These columns are making the wall of the microtubule. So how many columns are actually there? How many these filaments are there? This is called as longitudinal filament. This is called as longitudinal filament, or that is also called as protofilament. Protofilament. How many protofilaments are there in the microtubule? In a complete microtubule, there is presence of 13. There is presence of 13 protofilaments. 13 numbers of protofilament. This one is the one protofilament. This is one protofilament. So how many protofilaments? There will be presence of 13 protofilaments. Then that will be your complete microtubule. Okay. If you see here, this is complete microtubules, means there will be presence of 13 protofilament. But the B and C microtubule that are not complete, because there will be absence of whole 13 protofilaments, because this part of the B microtubule, this part of the B microtubule, that is contributed by the wall of the A, and this part of the microtubule, that is contributed by the wall of the microtubule, that is B. So this is not complete because there is absence of 13 protofilament. The 13 number of protofilaments that are absent here. Here also the 13 protofilaments are absent. Okay, and it is believed that there is presence of 10 protofilaments. There is presence of 10 protofilaments, and here also there is presence of 10 protofilaments. As the 13 are not present here, that's why that are not complete, that are incomplete protofilaments. Okay. So when we see the cartwheel structure, let's see the cartwheel structure of the centriole. How it will look. Cartwheel means just like the wheel of the cart, that is your sagar chaka, that is just looking like a cartwheel. So diagram of cartwheel structure of centriole. Okay, when we took the TS of centriole. Okay, so this is the middle part. This middle part that is made up of proteins. This is the center part, and this center that is called as hop. And from that center there is arisation of 9 spokes. This is 1, this is 2, this is 3, this is 4, this is 5, 6, 7, 8 and 9. Okay, so there is presence of 9 protofilaments, and these are joined with each other. Okay, so these are the this middle part, that is the hop, and these are the spokes. Cycle region, the spoke present. This is in the motorcycle or any wheel, there is presence of spokes. Like that, here also there is presence of spokes, and here there will be attachment of this triplet fibril. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. Okay, and here this is A, this is B, this is C, A, B, 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 C. Okay, and here the C microtubule is joined with the A microtubule of the preceding microtubule. Okay, here triplet microtubule. Suppose this is C, then the C is connected with the A, this C is connected with the this A, this is connected with this A, this is connected with this is connected with this is connected with this. So every C is connected with the, every C microtubule is connected with the A microtubule of its previous triplet microtubules, and this is called as what? This is called as CA linker. This is called as CA linker. Okay, so here there is presence of 9 triplet microtubules. Okay, and that are arranged in a cartwheel structure. That's why that is called as cartwheel structure of the centriole. This is just appear like a cartwheel. Okay, and this type of arrangement in case of your centriole, that is called as 9+0 arrangement, because the microtubules, the triplet microtubules that are only present at the periphery, khali bahar side these are present here, just these are present at the periphery. Outer side re, the peripheral side re khali present ache triplet microfibrils, microtubules, That's why that is the 9+0 arrangement. Okay, but when we see the flagella case re kana isi in the flagella case re amar in the middle re kana isi there is presence of microtubule, and periphery re sabu our microtubules one and the present isi, these are present in pair, microtubules are present in pair. Okay, so 9 1 2 3 7 8 9. Okay, flagella रे 9+2 arrangement देखबार के मिलसी, periphery रे 1 2 3 4 5 6 7 8 9 not a microtubules double condition dimer condition representation, and middle रे भी दुईटा presentation, so that is 9+2 arrangement, that is called as 9+2 arrangement. But here this is 9+0 arrangement because in the middle there is absence of any microtubule, that's why that is 9+0 arrangement. Okay, let me write something about this structure. So the cartwheel structure that is having when 3 microtubule join, they form triplet fibril, fibrils. Here a microtubule is complete, that is having 13 protofilaments, but B and C microtubules are incomplete because they are not having all 13 protofilaments. Okay, the wall of B and C microtubule is some part of the wall of the wall of B and C microtubule is contributed by protofilaments of their preceding microtubule. Okay, so in Centriole there is 9 triplet microfibrils. In Centriole there is 9 triplet microfibrils. Okay, and these 9 microfibrils are tilted in 40 degree in the central axis. These 9 microfibrils are tilted in 40 degree to the central axis, যদি আমি দেখো you see the structure इन्हें कहाँ होचे this is central axis है central axis जो दे रहा है central axis संगे there will be 40 degree. Okay, the corner will be the angle will be 40 degree here. That's why we are writing that 42 degree to the central axis. Then the 9 triplet fibrils are present at the periphery, not in center. Okay, that's why that arrangement is 9+0 arrangement. That's why that arrangement is 9+0 arrangement. Okay, then the triplet microfibrils are joined with C and A linker. The triplet microfibrils are joined with CA linker. Okay, and at the center proteinaceous rod is present called hop. In the hop have 9 spokes which joint the hop with a microtubule. Okay, a microtubule संगे join करो. That's why we are writing this. Okay, so this is all about your centriole part. So in this part or in this session, in this class, we have completed the cytoskeleton. Okay, the cytoskeleton. That is, in that session we have discussed about the microtubule, microfilament, intermediate filament, detailed structure about this microtubule, how this microtubule are arranged, and they are making the centriole, and what are the structure, what are the different parts of that centriole, what is that cartwheel structure of the centriole. Okay, so in the next class we will discuss about the last session or last part of our cell biology, that is microbodies. In the next class we will discuss about the microbodies. Okay.